Thank you very much, Veronica, for a quite extraordinarily kind introduction. And it's an enormous honour to be asked to give this 100th Galton lecture. Thank you very much for inviting me, to both you and Branwen. And I'm hugely enjoying this meeting. It's fascinating hearing about all these diverse fields. So as Veronica's indicated, I'm going to talk about ageing, and a lot of the impetus for work in the area is fairly obviously coming from the increase in human life expectancy that's been going on worldwide since about the middle of the 19th century. And it's illustrated here by the work of two demographers who've plotted life expectancy at birth, so that's a population average, against the year in which the birth took place for the country that was the world leader at the time. So initially this was the Scandinavian countries, latterly it's been East Asia, right now it's Hong Kong for both females and males. And you can see the extraordinary, inexorable 2.5 years per decade that's gone on ever since this trend started. And the point of this paper was the horizontal lines. It was the predictions of various individuals and organisations about where the trend was going to top out, what the intrinsic limitation on human lifespan was going to turn out to be. And the trend has gone past all of them. And these demographers were making the point that there's no signature at the moment in the demographic records, or at least there wasn't then, of where this limit would be. There was no approaching wall of death. And some of you who've been reading Nature and the popular press may have noticed that there's been some discussion about this conclusion recently, based around people like the world record holder, Jeanne-Louise Calment, French woman. She died at the age of 122 and a half in 1997. And this is a well-documented record. It's been checked because it was actually suggested that her daughter might have been substituted for her at a certain point, because there was a strong motivation for doing so because of the arrangements of paying her pension. But it does look as though the record's correct. And this group here confirmed that the trend in average life expectancy is continuing in most countries. But the point they were trying to make in the paper was that these outliers, these very long-lived individuals, and she died in 1997, no one has exceeded her record since then, and no one's close to it right now. So they were speculating that perhaps the real maximum has been seen. But I think partly for statistical reasons and partly for the kinds of reasons we've been hearing today, the changes in human circumstances. She was born in the 19th century. We have no idea what the environmental effects on the children who are in utero and who are born now are going to turn out to be in terms of their style of aging and how long they can live. So I think the maximum lifespan remains unpredictable. Something to be celebrated. We've improved living conditions, medical care, and so people are staying healthier for longer and living longer. But it's coming with the obvious downside that ageing's the major risk factor for all the nasty, chronic and killer diseases. In fact, I think aging should itself be regarded as a disease. These are just the plots for dementia, cardiovascular disease, and cancer for various EU and British censuses that have been done for the two sexes, and you can see this very nasty increase with age. So there's a lot of pressure from economic, health, just the point of view of personal suffering, to try to find ways simply of keeping people healthier as they age, not making them live longer, but compressing that morbid period at the end of life when things go wrong. And I think the way that a lot of people in this research area are starting to look at this now is that since the aging process is the major risk factor for these diseases, perhaps we might be able to find ways of intervening in aging itself to come up with a broad spectrum preventative medicine for several aging-related conditions simultaneously, rather than dealing with them piecemeal, as we tend to do at the moment, and as they arise. That might seem a rather naive way of looking at things, and we all know the aging process is very complicated. This is just a pictorial representation of a table that was produced some years ago by a pathologist, George Martin, working on aging in the US. And what he did was to simply list each human tissue system and what can go wrong in it during aging. And within each tissue, a number of different things can go wrong. And looking across tissues, there's no commonality in the kind of damage and pathology that accumulates. It looks as though each tissue is just accumulating problems in accord with the kind of insults of daily living that it encounters in accordance with its function as it goes through time. it seems very unlikely that any simple genetic or pharmacological or dietary intervention would be capable of capturing that loss and slowing it down simultaneously. And rather by the same argument, I think it was for a long time assumed that these laboratory model organisms, which have been so useful for other traits because of the very strong principle of evolutionary conservation, we can often take a gene from a mouse and put it into yeast or vice versa, or a human gene, and they function quite normally in the other animal. There's very powerful conservation of biological mechanisms. But these organisms have very different lifespans from each other, and they encounter very different kinds of stress and problems as they go through life. So why should there be commonalities in the way that they age? Of course, they're a rather non-random assortment of aging styles because they are laboratory model organisms, and there's a good reason for that. They're fairly short-lived, and also they have very high reproductive rate, which makes them easy to keep in the lab. Actually, most of the interesting variation in the rate of aging is out there in nature rather than in our labs. And these are just some of the heroes of that story, a fascinating collection of animals that live for a seemingly improbably long time. So this is the bowhead whale, which is the mammalian record holder. This Greenland shark turned up within the last 12 months. It turns out to be able to live for almost 400 years. This is from radiocarbon dating. It doesn't start breeding until it's 150. So it would be a lousy candidate for a fishery. It would go extinct immediately. A lot of animals, particularly ones that continue to grow once they've reached adulthood, so they've started reproducing, but they still increase in size, show negative demographic aging in the sense that they get more likely to survive and better able to reproduce for a period of their lives, as they are able to produce more eggs because they're big and also escape the attentions of more and more predators because of the size increase. I mean, it all comes down again at the end, but for at least part of life, we see this. This creature is absolutely fascinating. It's a eusocial mammal. It's a mammal that has sterile individuals within the social group that help the breeders with breeding. It's a rodent. It's intermediate in size between mice and rats, capable of living 30 years. Very interestingly, they don't get cancer. It seems to be something to do with the very strange nature of the extracellular matrix that produces this result, but they're completely cancer-free. There's no necessary correlation with size and lifespan, so this little bat, Brandt's bat, it only weighs 8 grams. This is somebody's finger that it's sitting on, but it can live almost 40 years in nature, and it doesn't hibernate. The correlation between size and lifespan can be very easily broken during evolution. And this is a rather quaint bivalve that lives in the North Atlantic. 507 years is the record. So extraordinary lifespans out there. But coming back to the rather more prosaic laboratory model organisms, they've told us some very interesting and important things about aging. The first of which is that the process is malleable. So this first came to light with just very simple chemical mutagenesis experiments with the worm, Caenorhabditis elegans. And what was found was that you can isolate strains of worms that have single gene mutations that make them long-lived. This is one of them here. These are actually data from my colleague, David Gems. And you can see the very nice increase in mean and maximum lifespan in these worms. This is a cumulative survival curve. This gene DAF2 actually turned out to encode the single worm insulin-like growth factor receptor from a previously unsuspected presence of this signaling network in the invertebrates. And interestingly, this effect of insulin IGF signaling turned out to be evolutionarily conserved in both the fly and the mouse. So these two have both lost a copy of the insulin receptor substrate. There's only one in the fly. It's called Chico. And you can see the nice increase in lifespan in flies that have lost either one or both copies of the Chico gene. This shows the IGF growth-like effect of the pathway. So if the flies lose both copies of Chico, you get slow growth to a dwarf adult. We know that the dwarfing and the lifespan are not causally related to each other at all. You can separate them. They're just two things that happen at different stages in the life history of the fly. This is a mouse that's lost both copies of the insulin receptor substrate 1. Again, a nice increase in lifespan. And one curious feature of these mutations is that they tend to increase lifespan very much more in females than in males, in both Drosophila and mouse, for reasons that we're not entirely clear about at the moment. We can't tell in the worm because they're hermaphrodites, but in these two, clearly the effects tend to be rather female-specific. And very importantly, this is not just an extension of the moribund period at the end of life. These animals are healthy and kicking long after the controls are dead. So, for instance, the worms and the flies remain much more mobile. They're better able to learn. They have better tissue function. They're clearly aging more slowly than the controls. With the mice, there's been some quite thorough phenotyping, and I think the really interesting thing about them is they show a very broad spectrum improvement in health during aging, just losing one gene. So although they start slightly insulin resistant when they're young, they look as though they might get diabetes as they go through life. They actually compensate, they grow their beta cells, and they therefore maintain their glucose homeostasis better as they age. They've got a better immune profile, more naive T cells, better rotor rod performance, they're more agile, they can hang on to a rotating rod. All of these are the same when they're young. These are phenotypes that kick in middle and old age. And they also show a delayed onset of specific aging-related pathology, so they get less osteoporosis. You can see the cataract here on the control. So these two mice are sisters. They're from the same litter, and they're about 850 days old here. And you can see the ulcerative dermatitis on the head and nape of the control. About 40% of them get it as they go through aging in this particular strain. We've never seen a case in the mutant mice, even if we high-fat feed them. So an interesting broad-spectrum improvement in health. There's obviously a lot of interest in knowing whether the signalling path or network is important in aging in humans. And the way that that's been tackled is mainly by looking at individuals or families who survive to advanced stages and asking whether they have particular genetic variants for the human orthologs of the genes that have turned out to be important in the animal studies. And also, they've been phenotyped for the expression levels of some of the genes in the signalling network. And these are just some of the results. The top two are genetic. So these are genetic variants in the actual insulin IGF signalling network that turn out to be associated with survival actually to 100 in this study, and particularly the IGF-1 receptor turned out to be important here. This FOXO3A is very interesting. So the insulin signaling pathway affects most of its phenotypes through the forkhead box-O transcription factor, of which there's just one in the genome of the worm and the fly. And that transcription factor is required for reduced insulin signaling to extend lifespan. If you remove it, reduced insulin signaling no longer makes the animals long-lived. And it turns out that variants in this particular one, there are 4 in humans, are associated with survival to advanced ages, 90 and 100. And then these are just gene expression analyses, particularly for the Leiden longevity study, which is looking at long-lived families, and finding that they show reduced expression of some of the key genes in this TOR pathway. So TOR is an intracellular pathway, and it's very intimately connected with the insulin IGF signaling network. And expression of its genes and also insulin-like growth factor again turn out to be associated with survival to 90 or longer in long-lived families. So early days, but it's looking as though it may be conserved to humans. So what we have here is a nutrient sensing network, both systemic and intracellular, very well known for its control of metabolism and for matching costly activities to the current nutritional status of the animal, so reproductive rate, growth, anabolic traits generally, now turning out to be important also in determining the rate of aging. And it's very well established now from animal studies that the network actually mediates the effect of a nutritional intervention, which has been known about for a lot longer, which is dietary restriction. So it was first discovered way back in the 1930s that if you simply force rats to eat less than what they would choose to eat if you left them to their own devices, so reduce their intake by 40, 50, even 60%, it produces a dramatic increase in lifespan. And because it's been known about for so long, those animals have been very thoroughly phenotyped, and they show an extraordinarily broad spectrum improvement in health. In fact, the only thing that or the two things that they turn out to be worse at than controls are resistance to certain viruses, so they're less able to clear an experimental flu infection. And they also have problems with wound healing. If there's any kind of trauma, then the animals have to be refed. And it's since turned out that the increase in lifespan in response to dietary restriction is extraordinarily widespread in the animal world. So it's been reported in all of these organisms that if they're put on a diet, lifespan's extended. So it's seen in worms and flies, also in yeast, actually, all the model organisms, and also a lot of non-model organisms as well. And recently, there have been 2 studies of rhesus monkeys, so very long-term study. These animals live 40 years in the US. They've been published in the last few years. This is actually a very interesting case of 2 different groups of people ostensibly asking the same question: does dietary restriction extend lifespan and improve health in rhesus monkeys? They actually did very, very different experiments from each other, which may explain some of the differences in their results. So one of these studies found an increase in lifespan while the other didn't. But what they both found was a very broad spectrum improvement in health. So these are just some of the things that they measured in the monkeys that have been published so far. These publications are still coming out. And you can see that in many different tissue systems and functions, the dietary restricted monkeys showed a great improvement in health. And one of the interesting things about dietary restriction in rats and mice is it's often much harder to say what a dietary restricted animal died of. It just died, whereas it's usually rather obvious with the controls that they've got a large tumour or some other failure of function that's killed them. So what I would like to do in this talk is to cover 3 topics that we've been working on in the lab, mainly in Drosophila, but also a little bit in mice. So I'd like to continue to talk about dietary restriction, and particularly the role of trade-offs, or the possible role of trade-offs in producing this increase in lifespan. I'll then come back to these sex differences that I mentioned for the insulin-IGF pathway, and then at the end just talk a little bit about how we might turn all of this into drugs. So first of all, dietary restriction and trade-offs. The way that dietary restriction is done in flies is just by diluting the food medium to which they're exposed. So they live on this gelled food medium. They basically lick the surface to get the nutrients. They lay their eggs on it. The larvae grow up in it. And it consists of sugar and yeast. And if you dilute the food, the flies don't respond by increasing their feeding rate for some reason. They just take it as it comes. And what you see is a very simple response, very similar to what's seen in worms and yeast, and also, at least for this half of it, in mice, which is that as you take down the food concentration, the lifespan increases through dietary restriction. If you overdo it, they fall off a cliff through starvation. And throughout that range, we see this decline in egg laying by the females. And what that's led to is a suggestion that over this dietary restriction range, perhaps it's necessary to pull back on producing eggs in order to get the increase in lifespan. Maybe there's a trade-off between them. In fact, there are some evolutionary ideas about this, that the whole response might be a mechanism for surviving intermittent periods of food shortage. What you do is pull back on reproduction, devote the resources to somatic maintenance, and produce the offspring under more propitious circumstances when the food supply returns. There's, as you can imagine, quite a bit of argument about whether that account of events is correct or not. But what's clear is that when you do dietary restriction, these 2 traits go in opposite direction from each other. So we became interested in whether there was a causal association between them, and the way that we tried to pull it apart was by looking at the nutritional basis of the responses. And as I've said, the diet consists of sugar and yeast, which just by coincidence have almost exactly the same calorie density as each other. But they have very different effects on lifespan if you take them out of the food. So if we've got fully fed flies here, if we take out calories by removing the sugar, the blue line, we see a very small response in lifespan up the y-axis. Whereas if we now take up the yeast, we get a much larger one. And this is the same shown doing it the other way around. So what this makes clear is that lifespan responds much more to the yeast in the food than to the sugar. And fecundity actually responds entirely to the yeast. It's not interested in sugar at all. So all of the action seems to be in the yeast component of the diet. And, of course, we wanted to know what it was about yeast. And this was looked at by 2 very talented characters in the lab. This is Richard Grandison, who was a grad student at the time, Matt was a postdoc. He actually now has his own lab at Monash University. We didn't have a defined diet at the time, so what we did was to take flies that were dietarily restricted. We measured everything that was present in the yeast and simply added it back to look at the effect on lifespan and fecundity. And it turned out that neither trait was in the least bit interested in the carbohydrates, the lipids, or the non-essential amino acids in the diet. All of the action was in the essential amino acids. And that's quite interesting because it's very clear also in mice, and there's some demographic evidence from humans, that the protein component of the diet is a particularly important determinant of health, both its quantity and its quality. But to continue with the flies, it turned out that for fecundity, methionine was the key amino acid. So what we have here are experiments where we've done the add-backs always with the controlled dietary restriction and the fully fed yeast on the right. If we add back all of the essential amino acids, we get the full fecundity response. However, if we leave out methionine, and only methionine, we don't get any response at all. For reasons I won't go into, we were interested in tryptophan as well at the time. It turns out not to be a player at all. The only thing that matters is whether methionine is present or not. Furthermore, over here, if we only add back methionine, we can get the full response that we get with essential amino acids or fully fed flies, and that's not true for any other amino acid. So for some reason, fly fecundity is interested in methionine. The story for lifespan is interesting and somewhat more complicated. The first point is that if we give them back methionine, which I've just explained completely restores the egg laying of the females, it has no effect whatsoever on lifespan. So with the methionine addition, we've got a fully long-lived, fully fertile fly with no trade-off between the two traits. If we do add-backs of more than one amino acid over here, the basic story is there seems to be a quantitative response to the number of amino acids that are added back. There doesn't seem, at the moment, to be any particular salience of any particular amino acid. So we were interested in why methionine for fecundity and what explains the lifespan responses to dietary restriction. And quite a team of us got together on this. So Matt, you've already met. Carlos Ribeiro has his own lab in Portugal, where he's very interested in neural control of feeding. Ming Yao and Sahar were both in Matt's lab at the time that we did this. And these three, George, Ilian, and Hannah, were respectively PhD student, postdoc, and intern in the lab in Cologne. And the idea that we came up with is, well, when a fly is producing eggs, what she's doing is basically producing more fly. So perhaps what she needs is the essential amino acids, the ones that she can't make, in the ratio that she needs them to make more fly. And we might be able to predict that by simply looking at the exome of the fly. So what we did was to simply translate the exome in silico in a computer using the genome sequence, and that gives us the array of proteins that the fly is making, and it's a short step from that to get the proportional representation of the essential amino acids. Now, of course, this takes no account at all of quantitative variation in the production of different proteins, which differ in their amino acid content. But for reasons we can go into afterwards if you want to, that actually seems to make very little difference. It doesn't seem to be important. This is a good enough prediction. So we've got the genome-wide average here, and by this time we've managed to produce a completely chemical defined diet for the flies. So what we did was to design fly diets that either contain the essentials in the ratio of the fly genome, or that were mismatched in two completely different ways, mismatch 1 and mismatch 2, shown here, with different increases and decreases in the different essentials. And we inquired whether the flies responded to these different diets in various ways. And the first thing that we looked at was their feeding preferences. We were interested simply in whether they could actually detect these differences. And some of these results are shown here. So we did experiments where we deprived them of amino acids, so just fed them sugar, and then asked them about their subsequent preferences. And the results of the choice assay are shown here. And it turns out that if we gave them the two mismatched diets against each other, they showed no preference at all. But if we gave them the mismatched diets, either of them, against fly, then they showed quite a strong preference for the fly-matched ratio. And we can ask the question in another way by pre-feeding them on matched or mismatched diets and asking them whether they're interested in yeast. And the results of that are shown here. So they can be pre-fed with either yeast or with the fly ratio, and in both cases they don't show a terribly strong preference between sugar and yeast. But if they've had a mismatched amino acid, then they show a significantly higher preference for the yeast. So the flies can tell the difference, and they clearly regard fly as more nutritious than the two mismatched diets. So we went on to ask, well, does that predict that methionine should be limiting for egg laying when we give them yeast? And the answer is yes. So this is the proportion of the essentials in yeast against the proportion of the essentials in fly, and you can see that methionine is the one that is actually predicted to be limiting. So we then went on to look at the effects of some experimental manipulations in the mismatched diet. So in the first mismatched diet, the limiting amino acid is actually arginine, with methionine as the next most limiting. So what we did was to experimentally test whether it would produce the predicted quantitative changes in fecundity. And the results are shown here. So if we start out with flies that are being fed with the mismatched ratio, and we add back 50% arginine, we get a nice increase in egg laying, as much as if we give them back all of the essential amino acids. This is just an arbitrary test with lysine. There's no response at all. And if we take these flies that have got all of the essentials added back and simply take out a third of the arginine, the egg laying comes back down. So it produces a nice quantitative prediction of their egg laying rate. Obviously, we can't go on doing that indefinitely, because sooner or later another amino acid is going to kick in as limiting, and in this case it's methionine. So we had a look at whether we could predict egg laying on that basis, and again it's shown here, same principle as in the previous slide. So we've got predicted egg laying in grey if only arginine is limiting, and you can see that it predicts that it will carry on up as we increase the amount of arginine in the diet. In fact, it tops out, and that can be predicted entirely if we take into account the limitation for methionine that kicks in after arginine has gone beyond the point at which it's limiting. And we did exactly the same set of experiments for the second mismatched diet, and it produced equally good quantitative predictions, somewhat to our surprise. We were interested also in whether this might be true in mammals. There are various reasons. The fly gut is simpler than the mammalian gut. It's possible that things that happen in the gut, and especially with the microbiota, might really alter things in mammals. So the first thing that we did was to go to the literature and find experiments where people had actually done nutrition experiments with rats and mice and found that particular amino acids were limiting for growth in their diet. And the results of some of these are shown here. And what we found in every case was that using the genome of the rat and the mouse and translating it, we could predict the limitation that had been seen experimentally by these other groups. So encouraged by that, we've moved into mice, and we've actually been looking quantitatively at effects on growth rate, for which I can show you the results. We're now also looking at the effects on adult health, for which I can't yet because we haven't got that far through the experiments. But these just show some of the effects on growth. So in most mouse diets, just standard chow that you buy from suppliers, the source of the protein is casein. And what you would predict with casein is that we've got tryptophan as the limiting amino acid. So we did some experiments where we gave the mice during growth a diet where protein was limiting for growth, and compared the growth rate either with the mouse amino acid ratio. This is using a diet where the amino acids are entirely crystalline. There's no other source of protein in the diet, but it's not a completely defined diet for the other constituents. And where they had casein. And again, we can quantitatively predict the difference in growth rate on these two sources of protein. So it looks as though it's a pretty good predictor of anabolism in the flies and the mice. And the result of that, coming back to flies and thinking about eggs, is that as we increase the amount of protein in the diet with these different amino acid ratios, what happens is that the egg laying, that there's an eventual physiological plateau in the number of eggs that a fly can lay. These are just our two standard diets that we use either for fully fed or dietary restriction and the level of fecundity that they produce. We've got the fly ratio in the middle, and as we increase the amount of fly ratio in the food, gradually the actual increase in egg laying sags off compared with the prediction, given the amount of the protein that's there, and it actually tops out at this 10.7 level, Whereas for both the mismatch ratios, the amount of the fecundity in relation to the amount of protein is still increasing at the higher ratio of 21.4. So what that means is that flies can achieve their maximum rate of egg laying at a lower protein concentration in the diet if it's matched to fly. And the consequences for lifespan don't seem to be very important. As I said, it just seems to be quantitative in terms of the different essential amino acids. So this shows the three ratios, the different amounts of protein in the diet. You get the standard dietary restriction of lifespan on all three, and they don't seem to differ very much from each other. But putting those ideas together, what it means is that lifespan and egg laying can be simultaneously maximised on an exome-matched diet, because here we've got the plateau of egg laying at 10.7 grams, and it's also the amount that maximises lifespan under dietary restriction. So you can completely get rid of this apparent trade-off between reproduction and lifespan by matching the amino acids in the diet to the amino acids predicted by the fly exome. So that was a lot of data and rather a lot of argument, so I've just summarised it here. If you match the essential amino acid ratio in the diet to the exome, it's a good predictor of anabolic traits. It also predicts growth in mice. Fecundity tops out at a nitrogen content on an exome-matched diet that also maximises the lifespan of the flies. So with an exome-matched diet, there's no trade-off between fecundity and lifespan. It's, in a sense, an artifact of having a mismatched ratio of amino acids in the diet. And this has some fairly obvious implications, I think, for the design of the protein content of diets in various contexts. And it would be interesting to know, for instance, if an animal's mounting an immune response and needing very large quantities of proteins with a slightly unusual amino acid composition, whether it would pay to alter the composition of the protein in the diet. I'll come on now to talking about sex differences in response to anti-aging interventions. And I will stick for the moment with dietary restriction because it's the one where we have the most data. And in flies, there's a very dramatic difference between the sexes in their response to dietary restriction. So this is the same plot that you've seen before of the concentration of the food medium. This is the response of the lifespan of females, very large amplitude response. Males respond very much less. In fact, in some experiments, they barely respond at all. There's some variation between runs. So we were interested in trying to pull apart what was causing that sex difference. And the people in the lab who've done this are two postdocs at UCL, Jenny Reagan and Mabina Chericha, who really got a grip on this one and figured out what was going on. And what they argued was, well, we're talking about dietary restriction here. The first thing that encounters the diet is the gut. So let's have a look at what's going on in the gut during aging. And what they unearthed was something that nobody noticed before, which is that there's a dramatic difference between the two sexes. And it's shown here. So we've got female guts at the top, males at the bottom. The flies are getting older towards the right. We're dealing with a columnar epithelium, just like in the human intestine. So if you look at young flies here of both sexes, what you see is this nice honeycomb arrangement. If you look down on the top of the epithelium, seeing the narrow ends of the cells neatly packed with each other. And if you take a section, there's this nice line up of the nuclei in a very well-ordered columnar epithelium. And as the females go through life, this starts to fall apart very quickly. So already at 21 days, which is the fly pretty much in the prime of life, you can start to see that there are cells missing, that there are clusters of stem cells. The fly gut contains somatic stem cells that give rise to the differentiated cells of the gut. They start to form clusters and mini tumours. And by 42 days, you're seeing these wound-healing rosettes. And by 63 days, which is a very old fly, the thing is a complete mess. And you can see this also in the cross-section. The gut just really falls apart as the female gets older. In the males, essentially, absolutely nothing happens. In a very old male, males don't even live as long as females, so this is a very old male. You can see that the gut still has this lovely, orderly appearance. So there's a huge sex difference here. And there are various other things that go with this obvious difference in histology. So I've said that the gut contains somatic stem cells. You can use the stain here, the Ph3 positive cells, to see them dividing. And you can see there's very much more mitosis in these middle-aged females. So they start out life with more stem cells, and they divide more actively as the fly goes through the aging process. We can also make a rather crude measure of the barrier function of the gut in the fly. So we can feed them a blue food dye, which in an intact gut just goes straight through. But if the enterocytes in the gut are starting to move apart from each other, which does happen during aging, then this dye leaks through into the fly, and you get a blue fly, which we call a smurf. This assay was actually invented in the lab of somebody called David Walker. So this is what they look like if there's a leaky barrier, whereas you only see the dye in the gut itself if the barrier function is intact. And you can see here in two different strains of flies that the females show a considerably higher rate of smurfing than the males do. Again, this is at 35 days in the middle of life. So it looks as though the barrier function of the gut also breaks down during aging in females. So what the two of them did was to go on and look at whether this pathology was to some extent rescued by dietary restriction. And they found that it was. So this is illustrated here. We've got dietarily restricted flies on the left and controls on the right, females and males. So all the action is in these older females. And you can see here just with an image from a single gut, so this is anecdotal, that they look much more disordered in the controls than in the dietarily restricted flies. This is a quantification here of the things that can go wrong. So you can get these small tumours, large tumours, or very severely disrupted epithelium. And you can see that in the fully fed females, there's very much more pathology than when they're dietarily restricted, nothing much going on in the males, as you would expect. So dietary restriction rescues this pathology. And that immediately raises the question, well, is the rescue of the gut pathology the cause of the sex difference in the increase in lifespan in response to dietary restriction? And one of the very nice things about flies for this kind of work is that their sex determination is cell autonomous. They don't have sex hormones, there's nothing systemic going on. Rather, each cell decides in accord with the ratio of the X chromosome to the autosomes what sex it's going to be. And the genetic cascade that affects that sex determination is very well understood, and you can manipulate it genetically in particular tissues using the usual tools that you can do with these model organisms. So what they did was to transdetermine the sex of part of the gut to ask if the dietary restriction lifespan phenotype went with the sex of the gut. And this is shown here. So what they've done is to feminize males. So they've turned part of the male gut into a female gut. It turns out for reasons that are basically technical, it's rather difficult to do it the other way around, which is actually the experiment that we really wanted to do first. And as a sort of control, they used a driver for the feminization that only feminizes the gut backwards from this point here, whereas this proventriculus, which is the part of the gut that produces those rather beautiful images that I showed you at the beginning of this bit, is left intact as a kind of control for whether the gut itself is okay. And so what we've got here are females, males, and the feminized males. This is the proventriculus, which isn't feminized, and you can see here that the males are just like normal males. Again, this is at 35 days, middle-aged flies. But for the R2 section, which is this bit here, they also looked at this section and it told a very similar story. You can see that in the feminized males, you've got this irregular arrangement of the nuclei in cross-section, just like in ordinary females, whereas the males are staying reasonably orderly, and whereas the males have this fairly even spacing of the nuclei, when you look down on the epithelium, the females are showing the disorder that we've seen before, and so are these feminized males. So we can transdetermine the pathology by transdetermining the sex of the gut. And these males in the feminized region of the gut also develop all of the other female characteristics. So if we look at the dividing stem cells, again, in the feminized males, they're greatly increased. Here are the ordinary males. If we look at the smurfing, it's also increased in the feminized males. And the same with the pathology. They show very female-like pathology in the gut in terms of the quantification of the different things that can happen. So, of course, what they did finally here was then to ask, well, what does this do to the responsive lifespan to dietary restriction? And this is shown here. So what they've done is to use just two levels of food, the one that maximises lifespan under dietary restriction and full feeding, just to keep things simple. And you can see here the dietary restriction response in the females in red, so the dotted line compared with the solid survival curve. There's a nice increase in lifespan. In this experiment, the males in green showed no response at all, which is not uncommon in these experiments. Males live less long than females, and we think the reason for that is there's a big difference in immunity between the sexes. Males have much less immune activity and are much less able to combat infections than females are. And this may be causal in their early death compared with females. So, of course, these feminized males now have a double whammy because they got to some extent, well, they've got their compromised immunity because they're male in all other parts of their body, but they've also got a feminized gut. So they live short, but now they show a very female-like response to dietary restriction. In fact, there's no difference in the magnitude of these responses. So it looks as though, at least in flies, the increase in lifespan in response to dietary restriction is largely because it rescues the pathology in the gut. We think the reason that the females get all of this gut pathology is simply that they have to eat an awful lot more. They're machines for making eggs, whereas males are just tearing around trying to find females. They just need energy. And probably it's this huge throughput and absorption of nutrients from the gut in the females that's making them have such a different kind of profile as they age. And at the moment, we're involved in looking at mouse guts to look at... Nobody's, oddly enough, ever looked at whether there's a sex difference in the way that the gut ages in mice, or indeed whether the aging of the gut is affected by dietary restriction. So it's something that we're looking at the moment. So finally, I'd like to say a little bit about drug targets. And there are a number of reasons for being particularly interested in these, and I've just listed them here. So one is that if we're ever going to translate any of this work into human health benefits, then it's going to have to be through pharmacological intervention. But for purely experimental work, drugs are jolly useful. It's much easier to do quantitative manipulations of the trait that you're interested in and of the target that you're interested in, and hence to get in this case, the dose of the drug that will maximize lifespan. It's much harder to do that genetically. It's easy to deliver or withdraw drugs at specific stages in life to ask about age specificity of the dose of the drug. And they can be very easily combined with each other. And probably for this nutrient sensing network, which we're so interested in, the one that mediates the effects of dietary restriction, these points are going to be very important. There's a lot of feedback in this signalling network, and it may be possible to stop it by going for more than one target. There's feedback at all kinds of levels, transcriptional, translational, post-translational. Drugs can sometimes, to some extent, cancel each other's side effects. And also, we've got strong reasons with this network for thinking that different bits of it actually go through different mechanisms. So this is just a cartoon of the fly insulin IGF-TOR signalling network. I mean, as in mammals, there are ligands. More in the fly, there are 7 insulin-like peptides. So it tends to be one of everything, where there are 4 in mammals. So single receptor, single insulin receptor substrate. And then the bifurcation of the pathway into the canonical insulin signaling pathway, which, as I've mentioned, goes through this fork-head box O transcription factor, or down the RAS branch of the pathway, which I'll come back to in a minute. And intimately connected with it is TOR, which is particularly involved in the sensing of amino acids through TOR in complex 1, mTORC1, which we know goes through these 2 routes. And we've got drugs against various bits of this. So rapamycin specifically inhibits TORC1. This drug, trametinib, which I'll say more about in a minute, is a MET kinase inhibitor in the RAS pathway. And lithium inhibits this gene product, which is called Shaggy in the fly. It's the fly GSK3. So rapamycin, to start with, is a very interesting drug. It's been shown some time ago to extend lifespan in flies. This was work of Ivana Bedov in the lab. She now has her own lab in the UCL Cancer Institute. You can see the extension of lifespan here. It's modest, but very highly repeatable, somewhat dose-dependent. And we know that the increase in autophagy and the reduced activity of the S6 kinase that are produced by inhibiting TORC1 are required for this extension of lifespan. But oddly enough, activation of 4-EBP is not. 4-EBP null flies respond perfectly normally to rapamycin. And there's a huge amount of interest in rapamycin in the field at the moment because it's been shown to extend lifespan in mice. So I think this is the most recent set of results on this from Rich Miller's lab in Ann Arbor, and you can see the dose-dependent response to rapamycin in males and females here. So again, it's sexually dimorphic. It extends lifespan more in females than in males, which is a bit of a hint it might be something to do with the gut. We don't yet know what the maximum lifespan extension is going to turn out to be because the maximum dose they used here produced the maximum extension. And because of this, there's been a huge amount of work on the mechanisms. And also quite recent interesting trial of rapamycin with dogs in Seattle. People were asked to feed rapamycin to their pet dog to look at various aspects of pathology as the animals got older. And they already actually found quite an interesting improvement in cardiac function in the treated dogs. But as you might expect for a drug that actually is tackling the underlying aging process, it's turning out to have much broader therapeutic effects than I think anyone suspected. So its licensed effects are mainly to prevent tissue rejection after transplant, particularly kidney transplant, to prevent restenosis after cardiac surgery, and as an anti-cancer chemotherapeutic. But it's turning out to do a lot more things than that, one of which is that it potentiates the effect of immunization in older individuals, both mice and humans. So mice are very like that, like us, in the sense that as they get older, they respond very much less well to immunization against flu. So this is just shown here with an experimental challenge with flu, and the immunized mice died almost as much as young mice that weren't immunized at all. But interestingly, in this study, what they showed is that if they pre-treated with rapamycin, so they gave them a course of rapamycin, took them off it, cleared the drug, and then looked at the response to immunization, now the old mice were way up with the young mice. They were showing just as much resistance to the experimental infection. And this turned out to be an effect on the hematopoietic stem cells. And this was actually picked up by Novartis, who did an essentially identical trial in older humans. And they found exactly the same thing, that the immune response to the challenge was greatly increased in humans that were pre-treated with rapamycin. So that was one previously unsuspected effect. Also, it's turned out to be protective in numerous animal models, worm, fly, and mouse, of neurodegenerative disease, particularly ones that are associated with toxic proteins. So this is one of the ones here. This is an amyloid beta, but the same thing's been shown for Huntington and for Parkinson's models. And it led to this, it's a really good read, this paper about the therapeutic range of rapamycin. So it's an interesting drug. We're also interested in this drug, trametinib, which is a MET kinase inhibitor. So one of the huge frustrations in the whole area at the moment is that there are no good drugs or chemical probes against the canonical insulin IGF signaling pathway. And if anybody in this room knows of one, I'd really like to hear about it, because so far nobody's shown that pharmacological inhibition of any of these targets can increase lifespan. But one of the odd things about the field is that although we all know that the RAS pathway is also downstream of the insulin receptor substrate, it's been roundly ignored in the aging field till quite recently. So because it's so important in cancer, about a third of human cancers contain a RAS mutation, there are many drugs against the targets here. And partly for that reason, we decided to have a look at whether it's actually important in the lifespan story, and it turns out that it is. So these two did the work, Kathy Slack and Nazif Halik. Kathy's actually just opened her own lab at Aston University. Nazif now has his at UCL. And what they did experimentally was to simply break the link between Chico and either the PI3 kinase, the canonical branch of the pathway, or the GRAB2 SOS-RAS branch of the pathway. And it was possible to do that because these insulin receptor substrates are really evolutionarily conserved. This is the structure of them. And it's very well known in mammals exactly which residues in Chico are important for the interaction with these other proteins. They're shown here. And already for the fly, as a result of the work of Maurice White, it had been shown that exactly the same residues are important for the PI3 kinase in the fly. And so Kathy particularly looked in cells and established that the same residue as in mammals is also important for this link. So what they did was to make flies that were null for Chico, and they put back either a genomic rescue to produce what should be the wild-type phenotype as a positive control, or a mutated version that couldn't talk to the PI3 kinase, or a mutated version that couldn't talk to RAB. And what they found was that nearly all of the standard insulin signaling phenotypes actually do go down the canonical pathway. So the slow growth, the small size, the oxidative stress resistance, the reduced fecundity, they're all canonical pathways. Lifespan's more interesting than that, happily. And it turns out that either branch of the pathway can equally extend lifespan to the full mutation. So this is just shown here. This is the rescue back to the control wild-type lifespan, the Chico mutant itself, and the two mutated versions. So both pathways are important. And not to go into too much detail, they go through different transcription factors, FOXO for the canonical, and E26 transcription factor called anterior open for the RAS branch of the pathway. And these two transcription factors certainly interact with each other. They overlap at about 60% of their genomic loci. And we don't know at the moment exactly what's going on there. The pathway somehow rejoins. But both branches are important. And that gave us a drug, which was this MET kinase inhibitor. So it's a GSK drug. It's fairly recently on the market, and it's against a particular stage in malignant melanoma. And it works very nicely against the fly MEK. So we can look at phosphorylation of its target, ERK, and we can show here that with insulin stimulation we normally see an increase, but in the presence of trametinib at a particular dose, there's no response, and it's quite specific. It doesn't affect AKT or the S6 kinase target. And if we look at the effect on lifespan of trametinib, we see a nice dose-dependent increase in lifespan. If we give them too much, interestingly, the young flies don't like it, but we still see an increase in maximum lifespan. So it looks as though an age-specific dose might be important. And finally, we're interested in this drug lithium, which inhibits the fly GSK3. And the reason we're interested in it is it's already been shown to produce a dose-dependent increase in lifespan in C. elegans, and in these different areas in Japan, there's an association between all-cause mortality and the amount of lithium in the drinking water. And Jorge in the lab has shown a dose-dependent effect of lithium in the fly. So we've gone on to look at combinations of these drugs. This basically shows that each one inhibits mainly their own target, with rather little crosstalk between them. And interestingly, they do turn out to be able to some extent, nullify each other's side effects. So as far as lifespan goes, rapamycin and lithium add on to each other very nicely. They each increase lifespan just as much in a fly that's already on the other drug as they do in a control. But down here you can see one of the side effects, which is present in mammals as well, which is that rapamycin increases adiposity. It increases lipid storage. Lithium on its own has no effect. Here's the increase with rapamycin. Lithium can completely reverse the adiposity. And when we add these drugs together, we can really get a quite spectacular increase in lifespan. So these are all the single, double, and triple combinations. And you can see the winner here is this triple combination of the three, a low dose of lithium, rapamycin, and trametinib. This is a 40% increase in lifespan, which is the biggest that we've ever seen in a fly. So I think what this is telling us with this network is that there are multiple targets within it that would need to be manipulated simultaneously if we want to maximize health. And I think what we're seeing in summary is the idea that we might be able to target the aging process to simultaneously protect against these age-related diseases. So it's a great pleasure to again acknowledge the people who I've mentioned as I went through who've done most of the work, also our funders, and you for your attention, and happy to take questions if there's time.
I forgot to say that actually we traditionally don't have questions at the end of this talk. Maybe you'd like to come to the front so that people can actually see you and see me presenting the Galton plate, which we present to each of our Galton lecturers. This is a silver dish that was designed by Leslie Durbin, who was born in 1913 and died in 2005. He designed the Queen's Head for the Royal Jubilee Medal, and the reverse of £1 coins in the 1980s. He also famously decorated the high-profile Stalingrad sword, which was given by Churchill to Stalin to honor the citizens of Stalingrad and their heroism in the 1940s. In 1982, he had won an exhibition in Goldsmiths Hall, and apparently his work is quite collectible. So this is